All right, so I'd like to introduce to you guys Dr. Marcia Hart. She is the scientific affairs and R&D manager for immunology at IDEX Bionerics. I'm sorry. She's going to be speaking about diagnostic testing methods for small mammals. Thank you, Marcia.
Thank you for that introduction. Hello, everyone. Thank you for attending. So today I'm going to be talking about diagnostic testing methods for small mammals. And see if I can get this to advance. Here we go. What I want to do today is do a little bit of background on sensitivity and specificity, just sort of as a reminder, because these factors are important when choosing a diagnostic test. We'll talk about various types of diagnostic tests. We'll talk about health monitoring strategies, and then, of course, sample collection and testing. So first, as a bit of background, it's important to remember that when we look at a group of animals, there's really no true ideal population. So in a perfect world, we would have a population of very discretely identified healthy animals and discretely identified diseased animals. But in reality, there's an overlap between healthy and diseased animals. And this is the area that is extremely important when designing a diagnostic test. So when we refer to this area, we typically talk about tests in the terms of sensitivity and specificity. Sensitivity is considered to be the true positive rate. It's the probability that a truly diseased animal will test positive. On the other hand, specificity is the true negative rate. It's the probability that a truly healthy, non-diseased animal will test negative. And this really matters when you're talking about testing, and especially assay design or how assays might be used between various diagnostic labs. So when we look at a healthy population and a diseased population, as I said, there is this little area here where they overlap. And this zone has a tendency to have false negatives and false positives. And where we set that cutoff point matters. So for example, if I am looking at an assay that has increased sensitivity, what I see is that if we move our bar here so that we have increased sensitivity, I'm going to capture all of the true positive animals, the truly diseased animals, but I'm also going to capture this gray zone, if you will, where there are animals that should be healthy and animals that are also diseased. So I have an increase in the number of false positives. What does that mean? So when I increase the sensitivity of the test, it means that I capture all of the sick animals, and we kind of see this by our curve, but more healthy animals will then be identified as false positive. Our specificity, on the other hand, is decreased. When I look at specificity, it's the converse. So when I set my assay cutoff to have a high specificity, I'm going to capture all of the true negative animals, but I'm also going to capture negative and positives in this overlap or gray zone. So what that results in is that I've captured all of the true negative animals, but I've also increased my false negative rate. So what does that mean in terms of testing? So when you increase the specificity of the test, we're going to catch all of the healthy animals, but we're also going to detect more diseased animals, and they're identified as false negative. And then in converse, my sensitivity is decreased. So why is all of that important? Well, as I go through the various different types of diagnostic testing, there are some advantages and disadvantages to each that are related to sensitivity and specificity. Also, as I mentioned before, sensitivity and specificity are important because it can also explain differences in test results between types of testing modalities and differences in test results between diagnostic laboratories. So let's shift a bit and talk about the different types of diagnostic tests available. When I think in terms of diagnostic tests, I think about tests that are direct tests. So I'm looking for the agent, or tests that are indirect tests, where I'm looking for some kind of response to the agent. So a nice analogy that I typically use is a tire and our tread marks. Our tire would be looking for the direct agent, whereas our tread marks is looking for the response to the agent. I know the tire was there by the presence of the tread marks. So examples of direct tests would be direct exams. So gross necropsy, just doing microscopy. Those are great exams. You could do culture, directly culturing samples from the animals. You could do histology, where we look at the agents and the morphology of those agents, and then our molecular tests, which are PCR-based tests. Our indirect tests, again, looking for response to the agent, are going to be serology, where we're looking for the antibodies to the agent, not necessarily the agent itself. Histology, where you're looking for some kind of lesion, again, not the agent in the lesion, but you're looking for some kind of a response, such as tissue damage or inflammation. Observation, so that's going to be clinical signs and lesions, and then a history and physical exam. So let's talk about direct exams, our gross necropsy. So gross necropsy has some great advantages. It's advantageous because it's rapid. I can quickly do a necropsy, assess the tissues, and have a general feeling for the presence or absence of disease. It's a great part of disease investigation. However, the disadvantages of gross necropsy is that it lacks the sensitivity. The problem with gross necropsy is that it is very rare to get those pathognomonic lesions that you see in the classic textbook, where you would be confident 100% that that animal has disease from agent X. So it lacks sensitivity. Histology for agent detection. This is a great method of examining for disease. The advantages are that it is specific and it detects active infection. So again, if I can find that ideal lesion during necropsy and I can sample that lesion, send it off to the lab for histology, it increases the chance that I can detect the agent within that tissue. The disadvantages is that histology for the specific agent lacks sensitivity. So if you think about, if you go back to your education days and you think about a microscope slide, those slides are very thin and narrow. In converse, if you think about a liver from a rabbit, per se, that's a very large organ. So when the histology lab takes that tissue, they take a section that you may have submitted for examination, and then they're going to take 5-micron sections and place on the slide. So if you think about the comparison of how big that liver is compared to how small that section is, it's very easy to miss the agent because of the size difference. So you're really looking for a needle in a haystack. So to increase the sensitivity, what people do is if that liver has a lesion, that's going to be the target area that you're going to submit for the sample. Histology is also quite labor intensive, as you can imagine, because you have to do the gross necropsy, collect the sample, formalin fix it, send it to the lab. The lab has to section it appropriately and go through all of the various staining techniques to get that sample properly prepared. There's also a lot of expertise required in looking at these samples and being able to distinguish, what is normal, what is a true positive finding for an agent, what might be artifact staining, those kinds of things. I put specificity as kind of a disadvantage because, again, it really depends on the tissue that you're looking at, the infection kinetics of the disease that you're looking for, the immune status of the animal. Is it immunocompromised versus immunocompetent? Those can all play a role in specificity. And then again, time, because as I mentioned, it takes quite some time to collect the sample, get it to the lab, and then have the lab do all of the various steps and have the pathologist evaluate the sample. We can also talk about histology for lesion detection. And this is a little bit different because this is more of a historical response. What I'm looking for here is the response to the infectious agent and not necessarily the infectious agent itself. The advantage is that it can be definitive if the lesions are pathognomonic. And as I mentioned before, that's really, really rare. But if that is present, if you have that exact great piece that you were able to identify during gross necropsy, and it has that classic lesion, that definitely increases the chances that you're going to detect and make a positive call. It's also really a great tool if you are working with anyone that's doing research studies. And the reason that it is such a great tool for research studies is because I know the infectious status of the animals. They typically are a control group, and then animals that are infected, or if you're doing a disease study, or perhaps they're infected and treated if you're doing some kind of a treatment type of study. Disadvantages is that histology for lesion detection can be less sensitive. And I typically think about this is in terms of viruses, especially when I'm talking about animals that are immune competent, because a lot of times those animals will clear the viral infection, and then you really don't see a lesion, or you see very minimal inflammation or changes. So those animals, you might falsely call them one way or the other. Other things too, disadvantages to think about is, again, labor intensive, the expertise that's required to look at all of these tissues, and then the time to just go through the processes of collecting and appropriately processing those samples for pathology review. Parasitology, it's a great direct exam. So advantages here is that it can be quite rapid. It usually takes a day or two to get those test results, depending on how quickly the samples get to the lab. It detects an active infection, so I'm not looking for historical infection here, I'm looking for active infection. It is quite specific, and depending on the species that you're talking about, it can be non-invasive or it can be invasive. Typically, when we talk about parasitology in our larger mammals and our larger rodents, it's non-invasive. There are groups that choose to use an invasive method for small mice and rats. That's typically done in vendors where they will submit a sentinel animal, and those animals are euthanized, and then various sections of the GI tissue are taken to evaluate for endoparasites. Disadvantages of parasitology is that it can lack sensitivity. Again, thinking through the whole process, did you collect the appropriate sample type? Was staff appropriately trained? Were they able to identify, the eggs or the actual agent itself? And then, as I said, there's lots of expertise and training that goes along with parasitology. Let's talk about culture. This could be bacteria, but it could also be for fungi as well. Advantages, it detects the active infection. It's usually specific, but there are some caveats to that, and that usually goes along with the various challenges with sample type, and we'll talk about that in terms of sensitivity, low sensitivity here in a minute. It is typically non-invasive, unless you're doing a diagnostic workup for an animal that currently has disease that you are going to euthanize, or perhaps died, and you want to do a diagnostic workup for those animals. But routine health monitoring is typically non-invasive. The disadvantages to microbiology is really that it has low sensitivity, and that really comes down to a lot of different factors. So one of the key players is fastidious organisms. So there's a little bit of an art to microbiology when you think about it in terms of some of these bacteria. You have to get the right kind of media under the right kind of growth conditions in order to grow whatever organism that you're looking for. And fastidious organisms are really quite challenging because for whatever reason, they typically don't like the culture conditions that we have. And so it can be challenging to get those organisms to grow. And so because of that, even though the animal may be colonized with those particular organisms, you may not detect it with culture methods just because they don't grow under the current culture conditions. Other problems with microbiology, or I wouldn't say problems, I guess I should say challenges, would be poor sample collection or storage. So a lot of organisms, especially organisms that might be oxygen sensitive, require transport media. And that's really to make sure that the organisms don't die during transit or are in low numbers, because that just makes culturing them so much more challenging on the laboratory side. The other thing too is storage. So if you're collecting samples for bacterial or fungal culture, it's ideal to keep those samples cold, not frozen, but refrigerated. And the advantage here is that if you freeze the samples, what can happen is that those freezing conditions are quite harsh. And so it can cause decreased growth by the time the sample gets to the lab. And again, that could result in false negative test results. Other factors to consider are wrong sample type. So, just understanding the pathobiology of the agents that you're trying to detect, so that you make sure that you take that correct sample type. Because if you take a wrong sample type, then again, you increase the risk of false negatives, not because the agent wasn't there or it couldn't grow, but really because we had the wrong sample type to test. Culture can be labor intensive because again, you have all of these different types of media. You have to make sure that those are set up appropriately. As I mentioned, you may have to use selective media, and choosing the right ones can, there's some time and training involved in that. And then of course, time, because you have to allow the bacteria or fungi to grow. Bacteria, we're looking at, sometimes you can have a rapid turnaround within a few days, but typically we let it grow out for several days just to make sure that we have adequate growth. And fungal cultures, if you can imagine those guys, they take much longer than bacterial culture. So there's a time component involved. When I talk about identifying the bacteria, how do we do that? So if you think back to maybe a microbiology class that you had in college, you think about those unknowns where you just got an aliquot of some kind of bacteria or a mixed population of bacteria, and you had a battery of media that you had to try to grow those organisms on. And then you had to look at the media and figure out, okay, did it produce a gas? Was there a color change? So typically what we've done in the past is use these selective media, do gram staining, and then do this biochemical characterization as I described in unknowns. And it's really an ID based on probabilities. And as you can imagine, it's quite subjective. So I'm looking for colors, change in color. I'm looking for gas production, all of these things which can be quite challenging. So several years ago, a newer method was developed, and that is the MALDI-TOF. It's the matrix-assisted laser desorption ionization time of flight, which is quite a mouthful, so we just call it the MALDI or the MALDI-TOF. And what MALDI-TOF is it's based on mass spectrometry, and so it uses lasers to ionize proteins or carbohydrates, various components of bacteria or fungi into an ion pattern. And that ion pattern, you can kind of think of it as a fingerprint for each organism. What you do then is you use that pattern and you compare it to a library of known previously identified organisms, and then you can identify the agent of interest that you're looking for. The process is that I need to isolate a single bacterial colony in order to do this. It takes just a few minutes, and then we get very strong IDs for most agents. Agents that we might not be able to identify because they don't exist in the library, there's other methods that can be used, but those agents are really quite rare. These libraries are very broad, and they contain a variety of human and animal agents. So moving on to serology. So serology is an indirect test because I'm looking for a response to the agent. The advantages here are it can be sensitive and quite specific, depending on where your threshold for the assays are set. It can be rapid. It's relatively inexpensive when you compare it with other modalities of testing that we'll talk about. It allows for both antemortem and postmortem sampling. And then the disadvantages here are really time. So it's a history of exposure. So in order to have an adequate antibody response, I have to wait for that animal to be infected with the agent and then mount recognize that agent and then mount an antibody response. And that typically takes between 7 to 10 days, maybe up to 14 days, depending on the age of the animal and that agent that you're talking about. Other disadvantages is that serology tends to have cross-reactivity. That can sometimes be an advantage because I could detect multiple viruses per se with a single assay. But typically cross-reactivity is troublesome because it can result in false positives. And agents that typically cause problems are going to be the bacterial agents, especially things in the GI tract, like Clostridial species are a great example of this, because there are a lot of similar proteins between normal commensal Clostridial species and Clostridial species that are pathogens. And so sometimes those antibodies you just sit down and on those you'll have antibodies to those commensal pathogens that will bind to the antigen in the assay, and then you get a false positive. The other thing to consider with serology is that it can't be used if animals are immune compromised. They just don't make an antibody response. So because serology is, I would say, kind of a cornerstone of traditional animal health monitoring, especially if you're just sort of monitoring groups of animals to look at health status, I'm going to spend a little bit of time on talking about the various types of serologic assays, because there are a lot of them out there and various laboratories are using them in different ways. So there are multiplex formats, which is kind of a new thing and is especially important in our rodents. There are ELISA formats, which are typically popular in our larger species. There are IFAs, HAIs, Western blots, and many other types of immunoassays. I'm only going to touch on these few because they're the most commonly used. So let's start at the beginning. So ELISA is an enzyme-linked immunosorbent assay. And again, we have a nice catchy nickname for that so that you don't have to say all of those words. It was introduced in the 1970s, and it really allowed for high throughput. The laboratory can push through many plates within a day, which is advantageous because we can get client results out quicker. The way that these assays work is that you typically have some kind of a plastic plate. So in this case, it's a 96 well plate. And then you bind an antigen to the plastic. I add my patient serum to that test well. And antibodies that recognize that antigen will bind tightly. Antibodies that don't recognize the antigen may bind a little bit, but it's not very tight. So what happens is when I do a wash step, all of these antibodies that are not binding to the antigen will be washed away. But because antibodies are not able to be visualized with the naked eye, we have to add some kind of a colorimetric reagent that allows us to detect whether or not they are present. So you use a secondary antibody that has some kind of a label on the end of it. It could be some kind of an enzymatic substrate, and it's an enzyme-related step, or more commonly in today's laboratories, it's a fluorochrome of some kind, so a fluorescent molecule. So I spend a little time here because all of the other assays I talk about, you will see it's a variation on a theme. It's all about antigen and antibody binding to that antigen and then detecting it. The part that varies is what is this antigen bound to. So in the case of an ELISA, it's a plastic plate. A second type of test is the indirect fluorescent antibody test, or IFA. The IFA is performed routinely as a confirmatory test. However, for some agents, it can be run as the primary assay. The way that this works is that you take an antigen and you place on a microscope slide. That antigen can be virally infected cells, it could be bacteria, it could be protozoa. There's a lot of different modalities. Again, similar to the ELISA, I add my patient serum, I add a detection antibody system, in this case fluorescent, and then I look at it under a microscope that has a fluorescent label or a fluorescent laser, a laser that excites the fluorescent molecule on that detection antibody. This allows the technician to be able to look under the microscope and see the results. So what you can see here are a bunch of cells. Here are nuclei and the cytoplasm. And you can see that there's a little bit of fluorescent background activity, but there's nothing that's nice and bright and has a structure to it. But when I look at my positive sample, I can see my nuclei of my cells. I can actually see, in some cases, division of the cells. So this is an individual cell, this is an individual cell. But what I think you can appreciate are all of these little dots in the cytoplasm. These are all positive cells that the antibody from the patient is reacting to viruses or viral particles in the cytoplasm. So this would be a positive sample. So this is what the technicians are looking for when they do an IFA test. There's also an HAI or hemagglutination inhibition test. This is used more for viruses than anything else. And it's used because some viruses naturally have this property of where they can agglutinate red blood cells. The red blood cells that are used are typically harvested from sheep. So it's sheep red blood cells, and then you add a virus, a known virus, parvoviruses, reoviruses, influenza are just some of the examples. And what happens is that the virus interacts with the red blood cells and makes this sort of lattice work, and it floats on the top. So this is a agglutinated sample. If I add my patient sample and my patient has antibodies to the virus, it inhibits this agglutination. So the virus can't sort of make this lattice work with the red blood cells. So the red blood cells just sink in kind of a pellet pattern to the bottom of the cell, of the well. So what I see here is that this is a sample where I would have antibodies to the virus and it inhibits agglutination. So I have this sort of clumping of the red blood cells. So usually what we do then is when we look at the plates, we're looking for antibody titers. So where does this inhibition stop? So you can see that in serum X, my patient stopped. So I didn't have antibodies that were able to inhibit agglutination at 1 to 40. So my viral titer then, or my antibody titer rather, is at 1 to 20. You can see this patient had a much higher antibody titer, and it didn't stop agglutination clear out to 320. So that's antibody titer for that particular patient. So this is how hemagglutination inhibition tests work. It's not as commonly used. Again, it's limited to viruses. And I don't know of many labs that are using it on the rodent side anymore, but I do know it is used on kind of larger species still. Then there's the Western blot. The Western blot is used a lot in research, but from a diagnostic perspective, it's typically used as a confirmatory test. It could be used as a tertiary confirmatory test after you've done some kind of basic serology, maybe an IFA, then you do a Western blot, or depending on the agent, sometimes a Western blot is the secondary confirmatory test. So how does this work? So if you think back to your basic biology and how proteins are structured, remember they have a primary, secondary, tertiary type structure. What we do is we treat them in some way, either by heat or by chemical treatment. We treat these antigenic proteins to linearize them. Once they are linearized, we place them in this kind of a gelatin matrix, apply an electrical charge, and then the proteins are then distributed according to molecular weight and charge. This gelatinous matrix then is the information from that is transferred to a membrane, which is, you can kind of think of it sort of like a paper. It's a nitrocellulose type paper. And that's what is the testable material. So we use the membrane. I add my client or my patient sample, and then I'm going to add a detection antibody system, and then I am looking for a banding pattern. So what we can do here is we can determine positive and negative status based on the banding. So here is a great example. I have my target protein at 25 kilodaltons. This was a positive sample, and these two would be considered negative. Here's an actual blot. So I have my mouse parvovirus positive samples, and you can see that there's very distinct banding patterns that are characteristic of mouse parvovirus of various sizes and weight. And then my MVM, which is another mouse parvovirus, but a different kind, you can see that it's negative for MVM. And then here are my negative control wells. So that's how Western blots work. So for small rodents, as you can imagine, a lot of these testing platforms use large volumes of serum or plasma. And in smaller rodents, that's just not practical. I just can't go out and grab a mil of serum from a mouse, for example. They're just not made that way because they don't have that much circulating blood volume. So what's been done is the development of these multiplex platforms, and they've really been a game changer in detecting antibodies in small rodents. So we have what we call planar arrays, which the platform is just flat, or suspension microsphere arrays. And I'll talk a little bit about each of these quickly. So they're just detecting antibodies to many antigens in a single sample. That's really the advantage. So rather than ELISAs where it's a single antigen in every plate, now I can detect multiple antigens, potentially hundreds, in a single sample. And that's why this is advantageous. So when we talk about planar arrays, it's just a flat surface. So here is a microscope slide. And on each of these slides, there's a grid pattern. And all of the dots within the grid is a single antigen. And that's what they're kind of showing you here in the illustration. So what happens is, similar to what we've talked about in all the other assays, you've got your antigen bound to a flat surface. I'm adding my patient sample and my detection antibody that has the fluorescent protein on it. And in this case, a computer with a laser is reading these slides and then generating data that allows you to be able to detect positive and negative samples. Again, here's kind of a similar platform. This is a 96 well plate versus the slide. Every one of these dots within the well is going to be a single antigen. So I think you can kind of appreciate the idea here that it's really multi-antigens in a single well or a single sample. Suspension arrays, same thing, but just using a different substrate. So here is a barcoded suspension array, and what these are little wafers, if you will, that have an embedded barcode in the material. These wafers are coated with antigen. I add my patient serum, I add my detection antibody, and then again, it uses a laser and a computer to read these samples. These are typically read not only with the laser detecting the detection antibody, but there's also a reader that detects the barcode. So in this instance, the barcode will tell you what antigen is on the wafer, and then the laser tells you whether or not you have antibodies bound to that antigen. Same thing with a suspension spherical bead array, but instead of having a barcode, now I have a fluorescent bead. So I have two lasers and two types of fluorescence. I've got the fluorescence of the bead, which identifies is what agent am I looking at, what is it coated with. It's coated with whatever agent of interest. You bind your antibody from your patient sample, and then you bind your detection antibody. So again, it's just a variation on a theme, but these suspension arrays allow you to detect multiple antigens in the same sample. So why do we like these? Again, it's a decreased amount of sample needed. So in ELISA, you typically need 25 to 100 microliters of serum, whereas in a multiplex, I can detect hundreds of antigens, and I only need 1 microliter or less of serum. So a single blood spot, 1 to 2 drops of blood is all we need. It's easy to test multiple antigens for the same animal. It's sensitive and specific. You can have multiple controls in the same well, which allows you to know, do I have enough immunoglobulin in the sample? Do I have great assay performance? Did I have good technician performance? Again, it just allows you to have greater control over the quality of the samples and the testing. And then you can do high throughput and automatable. So we can really run these through the lab quite quickly, which is great because then we can get the results out to clients more rapidly. Moving on to polymerase chain reaction, or PCR. This is a direct testing method, looking directly for the antigen, or rather nucleic acids of the antigen. The premise behind PCR is it really takes a small amount of specific DNA and it makes lots and lots of copies so that you can actually detect it. So that's kind of the whole story of how PCR works. There are many types of formats, conventional, real-time, multiplex, where you're looking at multiple agents in a single sample, reverse transcriptase PCR, where you're looking for RNA agents, particularly viruses. I'm going to spend a little time talking about conventional and real-time, just to give you some ideas of how this works. So in conventional PCR, the whole premise here is that you're taking a target molecule, you're going to make multiple copies by doing multiple cycles or repetitions. So the strategy for PCR is that you have a single tube of sample. You collect the nucleic acids from whatever sample is sent in, and then using heat, that DNA is kind of broken apart or unzipped, if you will, kind of like a zipper. And then what happens is my primers attach to the open strands, and then using an enzyme, it takes the nucleotides and just makes copies and copies and copies of the original template or the original DNA. Once the reaction is finished, I can use kind of a gel or a gelatinous material that has some kind of a stain or label in it to determine what's positive or negative, and that's the PCR product. So in this case, my sample is positive in my positive control, negative in my negative control, and you can see that animals 1 and 2 were positive for whatever this antigen is that we are looking for. So with conventional PCR, we really have to use this sort of secondary step where you have to use a gel in order to see what the product is. Newer on the line is real-time PCR. And really, the only difference with real-time PCR is that it uses some kind of incorporation of a fluorescent intercalating dye or a probe type system to really be able to look at amplification in real time as it happens. So in conventional, you have to wait for all the reactions to occur, and then you look at the product. But here, you can actually look at the product as it's going, or you can wait until it's finished. Because what happens is that during the PCR process, a fluorescent dye, or again, a fluorescent probe, this fluorescent reactivity is incorporated into all the copies of the DNA that you're making. So what happens is that the reader reads the fluorescent activity. And so what I can see is that here is my amplification cycles, and here is my fluorescent reactivity. And so what you can see is that with this particular sample, at cycle number 14, I had fluorescent reactivity. At cycle number 8, looks like at 18 to 20, it started coming up, so I had reactivity. So again, it takes away that need for a gel. You're just looking for the fluorescent amplification or the fluorescent incorporation during the amplification process. So again, PCR is looking for the agent of interest. The advantage of PCR is it's highly sensitive. You can take samples from multiple animals and you can pool them into one. It does have high specificity, so primers and probes can target unique genome segments, so that decreases background or things that I can't distinguish. It detects active infection when animals are directly sampled. So remember, PCR is a direct testing modality. You can use it for antemortem and postmortem sampling. Disadvantages is that it does have high sensitivity. So because of this, you can increase your false positive results. And typically that's done due to sample contamination, either during collection or during the processing in the lab. You can have false negative results. That's typically seen if you have the wrong sample type collected from the wrong location, or if there's inhibition of PCR reaction. Sometimes things like wood or different kinds of bedding components can inhibit PCR. Sometimes different types of feed components can inhibit PCR. They just inhibit those enzymes that cause the reaction to occur. And then other false negatives would be a sequence variation or a variant. So if I have some kind of mutation or some kind of change in the antigen itself that doesn't align with the sequence that I'm using for my probes during my assay design, we may need to go back and tweak or redesign the assay to be able to detect those variants. And then it can be expensive compared to other testing modalities. So for this, that kind of covers all of the different types of testing modalities. What I want to do is shift gears a little bit and talk about how to choose the right testing method. Because understanding disease pathogenesis is really key to knowing which of the methods that we just talked about you really want to choose in the process of your animal health monitoring. So for most agents, there is some kind of a shedding window, and if the animals are going to have an antibody response, there's an antibody response window. The shedding window for the agent can vary. It could be, if it's an immunocompetent animal, they may shed for a short period of time, antibodies rise, and then the animals don't shed anymore. Some agents are intermittently shed, so they may have a great antibody response, but for whatever reason, there can be intermittent shedding. Some agents, the animals are colonized for life, so they don't ever get rid of it, even though they may have an antibody response. Some agents have a poor antibody response. So this is just sort of in generalized kinetics, because I kind of want to talk about where or what sample types or what testing modalities you might want to use during what time period. So this is just kinetics of a rat parvovirus, and it's just comparing fecal PCR, which is here in gray, with serology antibody response, which is here in purple. And the point that I really want to make here is that if the animals, if you're bringing in animals from a new population and you want to make sure they're not shedding, PCR may be a great modality for you, because it's going to detect any animals that are shedding. And if you're out here in the later course of time, if you're not shedding, then those animals pose no risk to the population for this particular virus. If you want to have a historical perspective of whether or not this animal was ever infected with parvovirus, you would use an antibody response or serology test, because you can see they have quite a robust antibody response to the particular agent. The other part in here is that there is always a gray zone where there's overlap between antibody response and shedding. So depending on where those animals are in the kinetics, it can be important in how I interpret test results. So for example, if I'm only relying on serology to detect an agent, I may miss animals that are actively shedding depending on where I am in the serology window. So just something to be aware of. And like I said, I just use this as an example to illustrate the point that there are overlaps between viral shedding and antibody response. And it should be considered when you try to choose the right testing method for the purpose that you're trying to choose it for. Just a few more slides here. What are health monitoring strategies in small mammals? So who do I monitor? It really depends on your goals. You can test normal animals or diseased animals. You should really assess the risks to the population. Is it coming in through feed, bedding, new animals that I might be adding to the population? What is the goal of testing? Am I trying to prevent something entering the colony, or am I monitoring for existing pathogens? So on the prevention side, you may do quarantine, where you're really making sure anything you're bringing in is not posing a risk. Or you could do routine testing, just monitoring the health, making sure everybody stays negative. There are sentinel animal strategies where you use just a small population that is treated in similar husbandry handling to the large population at large, or you can directly test the population. You can use environmental testing samples, dust, water is a great one. A great example of that is with SARS-CoV-2. They're now doing monitoring of the human population by testing sewage water. That's a great example of how to do that by PCR. Monitoring existing pathogens, diagnostic workup of sick animals. That's a great way to do that. How many animals do I test? I hear this quite a bit. There's really no set number required for testing. More animals are required for agents with low prevalence. So here's a great pictorial of that. if my white animals are the healthy animals and my black colored mice are the diseased animals, as you can imagine, it's going to take sampling more animals to actually capture the diseased animals versus a colony or herd where there is high prevalence. So there's just as many white mice or healthy mice as diseased mice. I could probably sample fewer samples from this population. And then if I have a small colony or small herd, again, you don't typically need as many samples or need to test as many animals because the population is smaller. What animals to collect from? On healthy animals, you can do fecal pellets. Those are great for PCR testing, bacteria, viruses, endoparasites. You could do serum or dried blood spot. There's OptiSpot or the hematip microsampler that takes just a few drops of blood and allows us to test those for serology. You can look for antibodies to viruses. There's also some assays that do bacteria, respiratory fungi, and some intracellular parasites. So you'd want to work with your diagnostic lab to figure out, what assays might be available. You could do a fur pluck or skin scrape, pelt swab, or fur brush. And this can be used for PCR or parasitology to look for ectoparasites. And then environmental testing, you could do dust or other environmental components that might represent the accumulation of nucleic material from infectious agents over time. As far as sick animals go, samples from lesions are great. Those could be culture swabs for microbiology, could be a dry swab for PCR, could be a fur pluck or skin scrape if you're looking for fungal culture, or parasitology. Could also be done by PCR, depending on the agents of interest. You could do histology or a biopsy, depending on if it's antemortem or postmortem sampling. Serum or dried blood spot can be used, feces or environmental testing as well. And just sort of to wrap up here, so my key take-home points is really that you have to determine, why are you monitoring these animals in the first place? What are my goals? Am I trying to monitor something that's endemic in the population already? Or am I really trying to prevent something that's coming in? Am I doing a testing and cull strategy? What's the purpose of the monitoring? Once you determine that, think about the agents that you are worried about and what is the risk to that colony or herd. Could it come in with new animals? Could it come in with husbandry supplies such as food and bedding or water? Could it come in with my staff? There are very few agents that people do transmit to animals, but they do exist. So something to be aware of. Understanding that overall disease pathogenesis, all of these things are going to be really important in trying to establish what is the sample type that I need to take and what is the testing modality that I need to choose. And then really, if you get into any tricky situations or you just have questions on how these assays work or which one to choose or how to design a health monitoring strategy, reach out to your diagnostic veterinarians because I know they're very happy to help provide guidance in any of these different scenarios. And with that, I really want to thank you for your attention.
Okay, thank you, Marcia. Not showing any questions in the box. If you have any questions, put them in the question box and we'll get to them. Also, if you're on the phone, you can dial *9 and raise your hand and I'll unmute you. Okay, got a couple questions. Okay, are multiplex assays available commercially through Index for testing individual tiny pet patients, or is it more commonly used in larger scale laboratory settings?
Yep, I would say the answer is both. So we have them available for a variety of rodents ranging from mice and rats to a bunch of different species, all the way up from gerbils, guinea pigs, rabbits. So we have multiplex testing It is commercially used by large groups, but it's also used for small populations or pets as well. So usually I have people work with their clinical veterinarian who might be monitoring the population and submit samples for testing.
Okay, thank you. Next question, can you comment on the qPCR and its utility as a diagnostic tool?
Yes. It can be used. I think most of the time people are not doing like actual quantitation. We're really looking at more of a threshold type of a broad approach, but either method could be used. It kind of depends on the goal, because sometimes, like when I think about qPCR, I think about quantitating the result. I don't, as a veterinarian, I don't necessarily care if there's 100 copies. I just want to know, is it positive or negative? So, but if you ever have a question, most diagnostic laboratories will tell you, what is the genomic copy number for the assay that you're interested in.
Okay, thank you. Not showing any additional questions.